Good evening, guys. The video today is going to talk about kind of what we consider to be the central dogma of biology. And that is going to be the idea of going from our DNA to RNA to protein, because we know that our genetic information is contained in our DNA. And we need to take those genes and read them so that we can change them into protein form. Because when you physically look at somebody, it's not the genes that we see in them, it's the proteins that are being expressed. And so I have blue eyes, it's because the proteins that have been made to give me that blue eye variation. Of course, I have brown eyes, in case anybody ever paid attention. But again, those brown eye genes were read and then transcribed and translated into protein form. And so those proteins are what are showing up. That blue protein pigment or brown protein pigment is what is showing up in your eyes. And so we have to get from that gene to the protein. And so it takes place in two different stages. First is our transcription stage. And that's where we go from DNA to RNA. And then translation is going to take that RNA from transcription and it's going to change it into a protein form. And the thing to remember about these processes is that transcription is going to take place in a little bit different location depending on if you are a prokaryote or a eukaryote. And so transcription in a prokaryote is going to take place in the cytoplasm. And in a eukaryote, where is the DNA found? That's right, it's found in the nucleus. And so this process is going to take place in the nucleus of a eukaryote. Okay, transcription is ultimately going to end in the nucleus. And then translation happens in the same place for both. So in both prokaryotes and eukaryotes, we are going to find translation taking place in the cytoplasm on a ribosome. And so remember, because prokaryotes don't have organelles, then they can't obviously have transcription taking place in the nucleus. But we don't consider a ribosome to be an organelle because it's not membrane bound. So even though prokaryotes also have ribosomes where this takes place at, because they're not membrane bound, okay, we don't consider them to be a true organelle. Okay, and so we're going to talk in this particular video about transcription. And so for transcription, we're going to look at some specifics of it. Now, I know in biology, you probably just were told transcription is where we take DNA and turn it into RNA. It happens in the nucleus, and then that RNA goes to the ribosome, and it's translated by those, codon charts that you used. Unfortunately, just like with everything else in AP Bio, it's a little bit more detailed than that. All right, and so there's actually 3 steps that are involved in transcription. The first step is what we call initiation. Properly named, initiate means to start something. And one of the things we're going to see throughout this process of transcription is something that's called RNA polymerase. It should sound familiar because we had DNA polymerase during DNA replication. And so RNA polymerase is going to have a few different jobs in transcription. It's going to separate the DNA strand, because in order to copy DNA, just like we did with DNA replication, we're going to have to open that DNA strand up to make 2 sides of it, so that then we can copy those sides to make RNA strands. It's also going to be responsible for replacing U where we had a T. Okay, so we're going to have our uracil where we originally had thiamines, so that we can change from DNA to an RNA. Another thing we are going to do is we are going to attach to a promoter region, and I'll talk about this in more depth in just a second. And it's going to stop at a terminator region. Sorry about my kids in the background. And the last thing it's going to be responsible for doing, it's going to make an mRNA strand. The final mRNA strand that then is going to go to translation. So this RNA polymerase has a lot of jobs to do, but it needs some help. Okay, and it's going to need some help differently in bacteria versus in eukaryotic cells. And so with transcription and initiation, in bacteria or prokaryotes, in bacteria it's a pretty simple kind of system. If we remember our DNA strand has a 5' and a 3' on one side. On the other side, remember they run anti-parallel 3' to 5'. Okay. On a bacterial DNA strand, there's going to be a region on this strand, and it's going to be a series of bases, and this region is going to be called a promoter. A promoter region on a DNA strand is going to be kind of an initiation part of the strand. This is going to be where the RNA polymerase is going to start reading this strand to make an RNA copy. And so it's kind of like when we talked about in replication, we had those RNA primers that said, hey, DNA polymerase 3, this is where you need to start. The RNA polymerase is going to use this promoter region as an area to bind. And so it's going to recognize the promoter, and it's going to come in here, and I'm going to label this RNA polymerase, and it's going to attach there. That's where it knows to start the processing of transcription. And we're going to separate the DNA, okay, one side from the other. So we're going to separate it down the middle. And then we are going to replace U's with T's as we begin to make our RNA strand. Now, this is a side note. Remember in the replication video, I told you that DNA can only be made in 5' to 3' direction. The same is true for RNA. 5' to 3' is how RNA is made. So therefore, our template strand that we're going to use is going to be a 3' to 5' strand because we have to do anti-parallel. And so the bottom strand here is going to act as the template for our RNA strand that we're going to make. And so this RNA polymerase, it will go along in this direction. It will read the bases that are on the 3' to 5' side, and it's going to then make that new RNA model, or mRNA, which we'll talk about in just a second, and it's going to make that RNA strand in the 5' to 3' direction. That's called elongation. That's step number 2. We'll get there and talk about it more in just a second. But that's the overall picture of what's going to happen in bacteria in terms of initiation. So that's bacteria. In eukaryotes, it's very similar to what's going to happen, but there's going to be a little bit of addition of something. And this is one of my favorite biology words of all time. In terms of the DNA strand in eukaryotes, if we think of it being exactly the same direction here as our strand was above, we are going to have a promoter region as well. So this is our promoter region, we'll call it. And in our promoter region, you're going to notice it's a little bit larger because the actual promoter is located here. Before the promoter, and this here comes my favorite word, there is something that we call a Tata box. And that's what it's called, T-A box, a Tata box. And this Tata box is going to be recognized by transcription factors. And so we're going to have these things that come into play as well called transcription factors. And instead of the RNA polymerase binding like it did up here to the promoter, the RNA polymerase will come down and it'll recognize the Tata box. And now the RNA polymerase is going to bind to the Tata box, and it's going to recognize that as kind of its start place. That's where it's going to start reading the template strand, which again, the bottom is going to be the template strand. And so it's going to see the template strand and it's going to start reading it, and it's going to start bringing in the bases to match it to create, okay, our 5' to 3' RNA strand with our A, C, G, and U. Okay, so that is going to be our initiation for eukaryotes. Okay, so that's step number 1. Step number 2, which we've kind of already started talking about, step number 2 is going to be called elongation. And that's kind of what I showed you up here when I was talking about making the actual mRNA strand. And so with elongation, we've already had the RNA polymerase bind to the promoter. And so think of the promoter now being somewhere back here. And now our RNA polymerase, okay, is going to be down from the promoter. And coming out of the RNA polymerase is going to be that mRNA strand. And that mRNA strand is going to have its 3' end over here, its 5' end down here, and it's making and adding those bases to it as it goes. So that's elongation. Same process in eukaryotes as it is in prokaryotes. Okay? And the only thing that's different here is that in eukaryotes, we actually don't call this mRNA yet, we call it pre-mRNA. And we'll talk about that. And remember, the M stands for messenger. In prokaryotes, we are going to call this already mRNA, not pre-mRNA, because when we get to the third and final stage of this process, you're going to see that we actually have to do some modification to the mRNA in eukaryotes before it's ready to be used in translation. And so we call it pre-mRNA in eukaryotes at this stage. The third stage of transcription is going to be termination. It is aptly named termination because it is the end of this process. In termination, we are going to have the RNA polymerase is going to read a termination sequence. And so it reads what we call a terminator, which was very much like the promoter region. It's going to be a sequence that it's going to recognize that's going to tell the RNA to detach. So the RNA will detach from that DNA template, and it will release the pre-mRNA, or in the case of prokaryotes, just the mRNA strand, and the process of transcription will be complete. And so if we take again a look at our original DNA strand, here it was. The RNA polymerase is going to get to the end, to the terminator region. It's going to detach. And from that, we are also going to release that entire RNA strand that was 5' to 3' in nature. And so you're going to see it's just a singular strand of those RNA bases. Okay, and so termination is the end of the transcription process. But that doesn't mean we can go straight to translation. We can go straight to translation in prokaryotes. We can take that mRNA, take it right to the ribosome, and all is well. However, we need some additions when it comes to our strand in our eukaryotes. And so with our eukaryote modification, call it eukaryotic modification of pre-mRNA. So with the eukaryotic modification to our pre-mRNA, a couple things have to be added. And so in our pre-mRNA strand, if you take this as your backbone and these are your bases, as I've been showing you up until this point, and this is our 3' end, and over here is our 5' end. To the 5' end, we have to add a modification that we are going to call a 5' cap. This 5' cap contains some phosphate groups It also is going to contain some methyl groups that is going to protect it whenever it has to leave the nucleus. At the other end of the 3' end, we add on this group of just multiple A bases, and we call this a poly-A tail. Aptly named because poly means many, poly-A tail. And the poly-A tail is going to kind of do the same thing at the other end of the RNA. It's going to provide protection. And so the reason we go through these eukaryotic modifications is it's going to help the mRNA to get out of the nucleus and to the ribosome. Believe it or not, the cytoplasm is kind of a rough environment. In the cytoplasm, we have all these different enzymes that are looking to degrade things. And so when we have our nucleus and we have our RNA strand inside of it, it's protected. As soon as it goes out those nuclear pores and it leaves, it's now in an environment where it's not protected. And any enzyme could come along and try to degrade it. So what happens is that these poly-A tails and these methyl caps, the 5' caps, are going to provide protection so that we can't degrade this RNA strand, or we can't deteriorate it. It won't bind to an enzyme improperly and get destroyed. Another thing that it's also going to do is it's going to allow it to attach to the ribosome. The ribosome is going to recognize this 5' cap. It's going to recognize it as a kind of a signal that says, oh, this is my mRNA strand. I need to start translating at this point. And so it's another kind of helpful thing after that RNA gets out of the nucleus to get it to where it needs to be, to the ribosome. The other modification that needs to happen before we can call this specifically mRNA is we have to get rid of what we call junk DNA. So each transcribed RNA piece is going to contain things that we call introns and exons. Okay, so introns, and I'll give that an I, and exons, which are E's. Now, the interesting thing about DNA is that 98% of your DNA is introns. Okay? We also know introns to be called junk DNA. It's not that it's not useful, it's just not useful in coding. And so when we read a gene, introns are never really read. They don't code for anything. Exons are our coding regions. They are actually going to be important in coding for DNA. And so what we do before we take this pre-mRNA and make it into RNA is that we're going to have these exons and we're going to have these introns. And they're not evenly spread out like this. They can be in any combination. But we're going to take the introns and we're going to need to get rid of them. And we're going to splice together then those exons that are left. And so this is a modification that gets rid of that junk DNA that we don't need. To get rid of these, there's an enzyme that we call a spliceosome. Spliceosomes are going to be responsible for getting rid of these and taking these introns out. And so we're left with these exons. When we get to the unit on gene expression, another thing you'll notice is we don't always use all of the exons. And so sometimes we might get rid of this exon here and only use 2 of them. What does that do in the final gene reading? Well, that's going to change what is read. It's going to change the protein that is made. And so we talk about making proteins and changes in proteins. We can use the same genetic code, but take out different exons or put in certain exons to get a different protein being made from the same genetic code. So that's why we see variation among our proteins, even though it's the exact same DNA that we're always reading. Okay, so this is the end of transcription. Okay, we have modified our RNA to the point that it needs to be modified. We've added on that 5' cap. We've added on that poly-A tail. We only have the exons left that we need. And now it's going to be sent out of the nucleus into the ribosome for translation. So our next video will focus on translation.